So I want to switch gears a little bit and talk about learning and memory. This is always an interesting topic, but unfortunately, it's one topic that we still have a lot of unanswered questions about. But one of the things that I think is really cool is that a lot of what we know about learning and memory comes from a case study of someone known as H.M. We know now that H.M. was Henry Mollison, and interestingly, he's a nutmaker. He's from Manchester, Connecticut. Now, one of the unfortunate things that happened to HM is that he had to have his hippocampus removed. The hippocampus is a structure located in your temporal lobe. And when his hippocampus was removed, he had some changes associated with his memory. And this was really the first clue that this region of the brain plays a role in the process of learning and memory. But what's really interesting about HM's case is that his short-term memories were completely intact. So if you asked him to recite a word back to you immediately, he would have no problem doing it. His long-term memories were also intact. He could tell you about everything that happened before the surgery. He could tell you in a lot of detail about events that took place in his childhood. But what was observed by interacting with him was that after his surgery, he was unable to create new memories. So if you taught him a new word and you asked him to repeat it back to you a couple of days later, he would have no memory of that word ever being spoken to him. So there was this really interesting situation following the removal of his hippocampus. Some memories were affected, but others weren't. And so physicians and scientists started to subject him to a whole bunch of neurological tests. And there was one scientist in particular that he worked with, Brenda Milner, that had him do something known as a mirror drawing test. So a mirror drawing test you're seeing an example of on this slide here is basically when you look at an image in a mirror and then you try to draw it. And it's difficult to prepare for the mirror test because we're not used to drawing this way and you don't know exactly what you're drawing. So the first time that you do it, in everybody, you tend to make a lot of mistakes. And this was the same for HM. He made a lot of mistakes the first time that he did the mirror drawing test. But for most people, the next time that they do the mirror drawing test on the second day, on the third day, and every day after that, you start to get better at doing the test because you learn how to do the mirror drawing. And what was really interesting is that HM had normal results on this test. He got better just like everybody else did over a period of time. So this raised even more interesting questions about the hippocampus. Some memories, but not all memories, but now we have different types of memories as well. So he wasn't able to recall factual information over a long period of time, but he was able to learn new visual skills and new motor skills as well. And here's a video showing you another example of a similar situation in a man known as Clive Wearing.
One man is consigned to live entirely within the present, with terrible consequences. Clive Wearing has the worst case of amnesia ever known. 20 years ago, he lost his memory, and now his wife, Deborah, is the only person he recognizes.
Clive really only has less than 30 seconds memory, and sometimes it's as little as perhaps 7 seconds. It's as little as a sentence.
I'm going to see your sister, Adele. Her daughter's got married recently in New Zealand. And so they're having a party.
Funny how the ladies acquire a different title when they get married.
Do you know who I'm going to see tomorrow?
Buckingham Palace.
No, really guess.
I don't know.
You don't know? Adele.
Oh, I see.
Do you know why I'm going?
No.
She's having a party at her house tomorrow.
It's her birthday, isn't it?
No.
Yeah.
Do you know why?
No.
It's to do with her daughter.
I see.
Do you know why her daughter's having a party?
No.
Guess.
No, I don't.
She's just got married.
Oh, I see.
She just got married in... Do you know what country she just got married in?
No, I don't.
In New Zealand.
Oh, I see.
The sentence he is in, he will probably have forgotten the sentence before. You ask him a question and he'll give you an answer, but while he's giving you the answer, he's already forgotten the question. That's how short it is.
I'm going to see your kids tomorrow.
You want to see my kids?
Yeah, your children.
What are they up to now?
Do you know what they're up to now?
No.
Guess what you think they're up to.
No idea, I couldn't guess. I don't know what their O-levels were. They hadn't got an O-level last time I was conscious. So I didn't know what possible thing they could do, did I?
No.
No, I couldn't possibly guess.
Where do you think they are?
No idea. I don't know where I am.
Yep.
All he has is void behind him.
It's been like death. I've never seen a human being before. Never had a dream or a thought. The brain has been totally inactive. Day and night the same. No thoughts at all. As far as I'm concerned, the doctors have been totally incompetent. I've never seen a doctor the whole time. Oh, look, it's come.
So from studying HM and studying Clive Wearing and continuing the work in animals, what we know now about memory very superficially is that we essentially have two categories of memory. We have what we call explicit or declarative memories. That's consisting mostly of factual information, and that's stored in the hippocampus. So most of the information that you're learning for this exam, what you're trying to do is get that stored away in the temporal lobe in the hippocampal region, or in some cases just outside of it. But we also have what we call implicit or procedural memories. So that would be more like the motor memory that we saw in the mirror drawing task or playing the piano in the case of Clive Wearing. You also have some things that you just kind of know biologically from the time that you're born. And all of these things are stored outside of the hippocampus. And so they're not affected by damage to that structure. So what we know about the declarative memories and the explicit memories is that they're actually stored for the most part in the hippocampus. And we know that we have mechanisms that allow us to convert short-term memories into long-term memories. And so what we need to be able to do, and what most of us can do that HM and Clive Wearing can't, is do that conversion of short-term into long-term. So based upon all the research that's been done, it seems like there's some very specific things that we know are involved in that conversion process. The first thing is repetition. Most people have to hear or have to practice something over and over and over again in order to get it to convert from short-term storage into long-term storage. And that's one of the things that makes courses like this one so difficult. You often don't have the time to do that repetition. And so things stay in your short-term memory, but when the exam comes a couple of days, a couple of weeks, or even a month later, that information is essentially gone because it never made it into long-term storage. The other thing that seems to affect the conversion of short-term into long-term is the intensity of the memory. So something that you had a really strong emotional response to, like a wedding or a birthday party or a death in the family, most likely you'll remember the details about that event a lot more than you do the everyday minutiae of your lives, like what you had for breakfast last week. There's also a motivational component to learning. So if I were to give you a list of 10 words and ask you to memorize them, most of you would probably forget half of them, at least. But if someone were to put a gun to your head and say, memorize these 10 words, then more than likely you're going to be able to memorize all of those 10 words. So that's the motivational component. And that motivational component manifests itself in academics in that people tend to excel in things that they're interested in. And that's because they're a little bit more motivated to actually learn about them, even if it's subconsciously. But motivation's not the only factor. So sometimes we have a breakdown in our ability to form memories. For example, we know that memory formation decreases with age, and we also know that, interestingly, exercise can promote memory formation. All of this work was really launched by experiments by Brenda Milner in HM, and the person that's next to her in this photo, who's Eric Kandel, who won the Nobel Prize in Physiology and Medicine for his work on the molecular basis of memory. He actually has a really awesome Nobel Prize talk if you're interested in getting a crash course in learning and memory. But I'm just going to distill out some of the major components of his work here. So specifically, I want to think about the exercise component. And this video does a good job of summarizing some of the interesting experiments related to that. So this video really sets up the link between exercise and memory formation. Osteocalcin is a hormone that's released from bone, and it can travel to the brain and ultimately increase our ability to form new memories. Another example that you might be familiar with that drives memory formation is something called BDNF, or brain-derived neurotrophic factor. Now, what BDNF and osteocalcin have in common is that both of them work on tissues in the hippocampus, and they change those tissues at the molecular level. Now, memory formation is something that's really difficult to study in a laboratory setting, and it's especially difficult to study in animal models because it's difficult to test. And that's why we often have to use very complicated behavioral tests like running through a maze in order to understand memory formation. But one way that we can think about memories at a molecular level is in terms of graded potentials and action potentials. So when an electrophysiologist is interested in working on a memory problem, one of the things that they're looking for in their experiments is something called long-term potentiation, or LTP. If you think about what a memory is or might be at a molecular level, it might be a stronger response to a repeated stimulus. So on the left-hand side, we're seeing a presynaptic neuron here in purple and a postsynaptic neuron here in green. When the presynaptic neuron releases its neurotransmitter, initially, the green neuron produces an EPSP. Now, if no learning happens, then the next time you repeated this stimulation, what you would see is an identical response. You would produce an EPSP that had the exact same amplitude. But what usually happens when memories occur is you get a stronger response to the repeated stimulus. So in the image on the right, now what we're seeing are those same two cells, but when the neurotransmitter gets released from the presynaptic neuron onto the postsynaptic neuron, there's a stronger response. And if you look closely, you'll see that's because there's more neurotransmitter getting released onto the postsynaptic cell. You also have more ligand-gated channels on that postsynaptic cell to receive the message. So the response is to produce an EPSP with a bigger amplitude, but occurring in response to the exact same thing, the action potential in the presynaptic neuron. This is really the work that won Eric Kandel his Nobel Prize. It ultimately told us that at a molecular level, memory formation requires protein synthesis. So protein synthesis could refer to increased neurotransmitters. Protein synthesis could refer to increased expression of ligand-gated channels on the postsynaptic cell. It could even refer to the growth of new neurons or the growth of new axon terminals. So I might have more synapses between two neurons, or I might just have more neurons in general. Now, again, there's still a lot that we don't know about memories, but these are the two things that it seems like they have in common at a molecular level: stronger synaptic connections and more synaptic connections. And Eric Kandel showed that if you give an inhibitor of protein synthesis, then the mice never learn as they're running through the maze. It takes them about the same amount of time, regardless of how many times they actually are asked to travel through the exact same pattern.